Well, good evening, and welcome to tonight's lecture by Tom Schwan. Tom is a senior investigator at Rocky Mountain Labs down in Hamilton. He's received his BS in 1969 and his MA in 1972 in biology, both from California State University at Hayward. Between 1974 and 1976, he served as a Peace Corps volunteer in Kenya, a country that we heard about in the very first lecture by Tom Bolger in this lecture series. He received his PhD in medical anthropology, or medical entomology, in 1983 from the University of California at Berkeley. His dissertation investigated the ecology of fleas and plague in Lake Nakuru National Park in Kenya. From 1983 to 1986, he was a postdoctoral fellow and research associate at the Yale Arbovirus Research Unit at the Yale University School of Medicine, where he studied tick-borne viruses, and that's the subject of his talk tonight. He joined the Rocky Mountain Labs of the National Institute of Allergy and Infectious Diseases in Hamilton in July 1986, where he is now a senior investigator. While at Rocky Mountain Labs, Dr. Schwan has served as acting chief of the Laboratory of Microbial Structure and Function, the Laboratory of Human Bacterial Pathogenesis, and as the chief of the Laboratory of Zoonotic Pathogens from 2005 to 2013. He served on the editorial board of the Journal of Clinical Microbiology for 9 years, and he is currently on the editorial boards of vector-borne and zoonotic diseases and emerging infectious diseases. And he was an associate editor for ticks and tick-borne diseases. In 1993, Tom received the Award of Merit for Excellence in Technology Transfer from the Federal Laboratory Consortium for Technology Transfer for work related to the serodiagnosis of Lyme disease. In 2001, he was elected a Fellow of the American Academy of Microbiology, and he was selected as an ASM branch lecturer for 2011-2013. His research interests include medical entomology and the biology of ticks and fleas, the serodiagnosis of tick-borne infections, mechanisms of pathogenicity of relapsing fever and Lyme disease spirochetes, and how bacterial pathogens adapt for their biological transmission by ticks and fleas. And the other thing that's really interesting about Tom is he's the co-instructor of the course on vector-borne and parasitic diseases. That is one of the required courses in the minor in global public health. So some of you, I think, have had him as an instructor. So I think you're going to find his talk really fascinating tonight. So please join me in welcoming Tom Schwan.
Thank you, Peter. I think I'm mic'd up, so can folks in the back hear me okay? All right, great. So I'm aware of the time. I'm sorry I gave Peter such a long introduction. I should have kept it shorter, but thank you. That was very nice. So my goal here tonight is talking with a group I don't know who has a diverse background that I don't know, to hopefully have you leave here with one, enjoying the talk and having an appreciation for a tick-borne disease that now is established in western Montana. And I'll do a little comparison to a similar type of disease that's occurring in West Africa, where we also have been working for the last 8 years. So I have a lot of pictures. I go quickly. I don't like the screen behind my back, but I'm going to do the best to make this interesting and enjoyable for you. I just want to start by thanking all the folks that have been working with me over the years. I'll name and picture some of these folks specifically. I don't have time to go over their entire list and what they do, but I've been very fortunate over my 28 years at Rocky Mountain Lab to have a very talented pool of postdoctoral fellows, technicians, students, student interns. And all the work has been supported by the National Institutes of Allergy and Infectious Diseases. Here's one picture of one of my groups, one of the more recent ones, and if you just go from left to right, I'll just go real fast because you'll hear their names: Paul, Sandy, Usman, Marie, Bob, Tammy, Brandy, Tom, and Colleen. All right? One thing I want to stress with you, and so when you leave here, you'll have appreciation of how wonderful ticks are compared to all other arthropods. And Peter's shaking his head. Now, if you look at this list, what I've got are things that you probably have all been bitten by. Mosquitoes, black flies, maybe not sandflies, horseflies, midges, probably not tsetse unless you've been in riparian African areas, and fleas. And fleas are less of a problem here than they are in many other parts of the United States. These are all obligate blood feeders, but only as adults, and for some of these groups, only as adult females. Right? Only adult females. Tsetses and fleas, the males and females feed on blood. And then for these insects that have a incomplete metamorphosis, all of the stages feed on blood, but they're relatively short-lived. All right? So mosquitoes, it's only the females that feed on blood, and they don't live very long. Now, if we compare that to ticks, ticks are also blood feeders. They're obligate blood-feeding arthropods in every stage of their life cycle. They do not feed on anything else. They have to feed on blood. They take a lot of it. They live a long time. And because of their long-lived and their morphological characteristics and their obligate blood-feeding behavior, they are very important arthropods to acquire infectious agents in their blood meal, which then, if they can propagate that organism in their body, they can subsequently feed and transmit it to the next host they feed on. So ticks are really important blood-feeding arthropods as what we call vectors for agents that can cause disease in humans and domestic animals. Here's a little more detail about the two types of ticks. We've got hard ticks and soft ticks. You're more familiar probably with the hard ticks like we have out here right now. It's tick season for wood ticks in the western parts of Montana. This is the Dermacentor andersoni, the wood tick, that overwinters as unfed adults. When the snow melts, the sun warms up the southern-facing slopes in the canyons. The ticks come out. They put their legs out. They wait to be picked up by an elk or a deer or a moose or whatever, and they feed for 6, 8, 10 days, suck that blood, make eggs, reproduce, and die. All right? So in the hard ticks that are on the left, they have a 3-stage cycle. They all feed on blood. Larvae feed on blood on mice. They molt to nymphs. They feed on blood on small mammals. They molt to adults. They feed on larger mammals, reproduce, and die. The ticks on the right, the soft ticks, are the ticks of my passion and career. These ticks are secretive. They have multiple nymphal stages, not just one. And once they reach adulthood, they just don't feed once and die. They feed and lay eggs. They feed again, lay eggs, feed again, lay eggs. This is what they call a strict gonotrophic concordance. One blood meal, one egg batch. All right? And these ticks can do that for years. So they have, over the years that they can live and feed repeatedly and reproduce and lay eggs, they can have a high reproductive potential. But what's interesting about these ticks is that they're secretive, they're fast feeders. The ticks on the left will attach and feed for 6, 8, 10 days. The ticks on the right will climb on at night. They're nocturnal. They suck blood for 30 minutes and get off and go back to where they came from. All right? So they're stealth feeders. Most people, when they're bitten by these ticks, they don't even know they've been parasitized by them. And between feedings, these ticks on the right can sit and fast for 1 year, 2 years, 5 years. I've got some that have been in the lab that haven't fed for 10 years, and they're still alive. So these ticks on the right, forget the ones on the left. They're really kind of boring. All right? These ticks on the right can far outlive the vertebrate hosts that they feed on, and therefore, if they are vectors of infectious agents, they are the true reservoir, not the squirrels or the mice or whatever they feed on. All right. So this is a deer tick that transmits the Lyme disease spirochete. That's a nymph before feeding and 3 days later. So you can see they get quite large, but it takes 3 to 5 days. This is the tick that we work on, Ornithodoros hermsii. This is the vector of relapsing fever. That's the same tick before and then 45 minutes later after it engorged on a mouse. And then they climb off and go back to where they came from. And the ticks have beautifully adapted terminal mouth parts so that they can cut the tissues into the host. They've got to get in there to get that blood flowing. So they push and cut, and then they spit. They've got saliva that has hundreds of pharmacologically active compounds that keep the blood flowing and alter the cellular components of the blood, because mammalian bodies have a defense to stop blood from oozing out of them if they're cut, all right? You know, clotting mechanisms, platelets, etc. And ticks in their saliva have mechanisms to keep that blood flowing. Fascinating biochemical reactions. So they can cut, pull, spit, cut, pull, spit, and have a successful blood meal. Now this is the tick that's around here right now. It's tick season. This is the Rocky Mountain wood tick. We have a 3-month window of opportunity for you to be infected by by these ticks. Spotted fever is the most serious. 1% of the ticks on the west side of the Bitterroot Valley have virulent spotted fever rickettsia. People die every year of spotted fever. These ticks also carry Colorado tick fever virus. It's not a lethal virus, but it has a higher prevalence in the ticks, and so every year in certain canyons there can be 5 to 15% of these ticks infected with this virus that can transmit to people. And then there's other things like Q fever, another bacterium, rabbit fever bacterium, Francisella, that's another bacterium that's serious. These ticks can also cause a paralysis. A single female tick can climb on you after 5 days of feeding, can cause a fatal paralysis. Bottom line, you don't want these ticks to feed on you. All right? There's a lot of serious health outcomes if you let these ticks feed on you. All right. Now, the rest of my talk is going to focus in on something that I just love working on. The disease is called tick-borne relapsing fever. And it was first found in North America in Colorado in 1915. So I've got an old map here from a 1977 publication showing the spot where the first 5 cases were identified in Colorado. All right, and I'll explain how these were identified. What you will learn when you walk out of here is the basics of relapsing fever. These dots show locations of various tick species, and the shaded areas show areas which are considered endemic for where people can get infected with these bacteria that cause relapsing fever. This map now is way out of date. The main thing I wanted to show is first cases, 1915, Colorado, and in that paper they didn't even mention the role of ticks in transmitting this infection. Okay, so here's one synopsis of information that for the students, whatever you're doing, you should know. This is a tick-borne zoonosis. What's a zoonosis? That's a disease of humans that results from the infection of a pathogenic agent that's circulating in a wild animal or domestic animal population. All right? If someone gets a zoonotic infection, they're not getting it from their wife. All right? They're getting it from their cow, their dog, the mouse running out back, and you can get these things in a variety of ways. Virus in urine, contaminating the food, tick bite, flea bite, mosquito bite, all right? So there's a variety of vector-borne diseases in North America that are zoonotic agents, and other vector-borne things may not be a zoonosis. Malaria is a parasite transmitted from one person to another via mosquitoes. Even though it's mosquito-borne, it's not a zoonosis. The parasite's not maintained in a wild animal cycle, okay? So relapsing fever is a disease caused by spirochetes, a bacterium circulating in nature, maintained between ticks and small mammals. And when people enter into that maintenance cycle in a particular ecological setting, they can be fed upon by those ticks and get infected and develop the disease. There's no vaccine, but it's easily treatable with antibiotics. And the way a diagnostic lab identifies these cases is by making a thin blood smear when the patient is acutely ill, staining it, looking at it under the microscope, looking for the spirochetes. That's how this infection is confirmed in the laboratory, the visualization of spirochetes in a stained thin blood smear. The most important clinical outcome of this infection in North America is if a pregnant woman gets infected, it can kill her fetus. All right? It's rare, but the last time it happened was in Spokane in 2000, and here we got the blood at the lab. So this is a thin blood smear that I stained by immunofluorescence with a monoclonal antibody. This is a massive number of spirochetes that were in this child that was delivered by cesarean birth, and the baby died 9 days later. All right. Another list of critical information for you to walk out of here for relapsing fever. All right? Most cases of relapsing fever in North America are caused by one species of spirochete. It's called Beryllium hermsii. That's what we work on in our lab. Most people, when they get infected, they're not in home. They're in a recreational cabin in a higher elevation coniferous forest where there's chipmunks, squirrels, ticks, and spirochetes all coexisting in this recreational mountain setting. When people sleep in those cabins and the ticks start wandering at night, they can feed on people rather than the squirrels, which are diurnal. That's part of a little bit of a problem. All right? So in the cabin is where people most often get fed upon. It's a rare disease, but there have been outbreaks. Years ago, there's a big family reunion on the north rim of Grand Canyon. 45, 50 people got sick. They all went, and the characteristic of this disease, people go somewhere, they get infected, then they go somewhere else and get sick. And that complicates the diagnosis. All right? They don't usually get sick at home. Yeah. All right. And as I said, well, I want to mention that these diurnal exposures, we've worked up a few cases where people actually got infected, not while they were sleeping, but by what they were doing during the day. And just a few months ago, we were able to report for the first time that this spirochete causes a disease in domestic dogs. So that was the first time that's been identified. So it's kind of fun. Okay, here's the distribution of Beryllium hermsii in Western North America. This is my map as I currently have it. The blue area sort of shows the perimeter within which the ticks, the right mammals, and the spirochetes can co-occur to give rise to human infections. Now, you can't find these components, tick on the left, pine squirrels, chipmunks on the right, everywhere within that green spot, right? You've got to have higher elevation mountains with the right trees, with the right food sources, etc. So I've got this big, funny shaped blue area, but relapsing fever does not occur everywhere within that. You've got to be in the right ecological setting where you've got the ticks feeding on the pine squirrels and the chipmunks and actually have the spirochete that's being circulated between them, so that when a person enters into that specific ecological cycle, gets fed upon at night, they can get infected. Now I've got white and dark dots up here, which mean from our isolations of the spirochete from people primarily over the years, we've been able to distinguish genetically two types of spirochetes. And that's really not of any importance for you folks, but for us that work on it, it's kind of cool. So depending on the DNA sequence, we'll call the bug either genomic group 1 or genomic group 2. So, and you can see, and I'll talk about this in a minute, up at Flathead Lake, we have spirochetes that have been genetically typed to both genomic groups. Okay. So the key components for Beryllium hermsii, the bug that most frequently causes relapsing fever in North America, has the vector Ornithosaurus hermsii and small mammal vertebrate host to maintain the spirochetes in nature. Okay. I just mentioned that by DNA typing these bugs, we see two genetic types. Again, for us, that's interesting. For you, it's probably boring, but I'm going to make it interesting, hopefully, in a couple of minutes. So this is just taking one gene out of the genome of the bacterium. They've got about 1,500 genes. Sequence one gene and compare it from all of our isolates, and the groups fall into two distinct groups. All right? And we're about to probably name one of the groups a new species, but I've been really hesitant to do that. Okay, I mentioned that you diagnose the disease in a human patient by doing a thin blood smear, staining it with the right Giemsa stain, putting it under the microscope and looking at it at 600 or 1000x oil immersion. And hopefully you can see those spirochetes even in the back. In the back, can you see those little squiggly lines? That's what you look for to confirm that a patient has tick-borne relapsing fever. Right? Now, I'm just going to say, you can't do that for Lyme disease. I really don't want to talk about Lyme disease tonight, but you cannot confirm a case of Lyme disease by making a thin blood smear and staining it, because the spirochete that causes Lyme disease never achieves cell densities in the blood that are high enough for an observer in the clinical lab to see the spirochetes with a microscope. Can't happen. You don't diagnose Lyme disease by looking at a blood smear and looking for spirochetes. Right. Now, relapsing fever. Why is the name relapsing fever? These spirochetes have the genetic capability of antigenically varying their outer surface proteins. So that, if we just look at, let's just look at right here. All right? This shows increases in spirochetes with days in a mouse. If you see these spirochetes during that peak, almost every spirochete in that population has the very same outer surface protein. All the same. Enough so it can stimulate the host to make an antibody response to that population of spirochetes. And they get cleared. They get killed by the host antibody response. But there's a rare antigenic variant within that population that did not stimulate an antibody response and then is immune to the antibody attack that's generated to that first primary population. So in this population, or in this population that produces an antibody response to it, there's a rare variant that isn't killed, and with time and over days can repopulate the blood to a new spirochetemia, and now the person gets sick again. All right? Then there's another antibody response to it, and they get cleared. But because of this genetic mechanism to vary antigenically, the spirochetes are able to sequentially repopulate the blood, get cleared, repopulate the blood, and hence the name relapsing fever. All right? That person gets sick, they get better. They get sick, they get better because the bacteria are growing, they get killed, they grow, they get killed, they grow, they get killed. All right. All right, in 2002, an interesting thing happened up at Flathead Lake. So I imagine all of you know where Flathead Lake is. It's an hour north, right? All right. It's the largest natural freshwater lake west of the Mississippi. And it's got a large island there, Wild Horse Island, which is now a state park for day use only, but it used to be privately owned, and so there's a lot of grandfathered in private properties. Okay, here's a Google map of it. And so it's got yellow pine forest here and over here and native grasslands. And then around the perimeter of the island, and have any of you guys been out there? Yeah, yeah. So you know what it's like out there. The lots are circular, of either 1 acre or ½ acre size. A lot of the lots have cabins, and it's there's one guy lately that's been out there all year, but usually it's just there's nobody that usually lives there out all year round. But it's a beautiful spot. 2002, there's a family reunion at this cabin, and the first family that got there decided to do some cleanup before the other 18 people arrived to have the family reunion. So this is a large cabin on the south side of Wild Horse Island. And what happened was a guy got sick. He was so sick he went to Polson. They told him he had a virus and sent him back to the cabin. He got better. He was really sick, but he got better. He had a nice family reunion with 20 other people, and then he started driving home to Seattle, and he got sick. He got really sick. So his wife drove him right into right to a ER of a hospital in Seattle. And then that night I got the call from the ER doc and the infectious disease guy and said, we have a guy that just got back from Montana, and he was out on an island, and you know, he's really sick, and he's now sick for a second time. Do you think he could have relapsing fever? And I said, well, yeah, I mean, so, of course, what do you think I asked him first? Blood smear. I said, exactly. All right. Did you make a blood smear? Did you look for spirochetes? Yeah, but we didn't see anything. Okay. Well, we can look for antibody in a couple weeks. Yeah, let's just see how it works out. 2 days later, I got another call from the same physician. Another family member in Seattle was now also in the hospital and acutely ill, and there were spirochetes in the blood. So they had spirochetes in the blood. And then a cousin that flew back to St. Louis, Missouri, became suddenly ill with spirochetes in the blood. Remember this? So people were getting sick on an island in Flathead Lake but they were coming down with the illness and being examined in Seattle, St. Louis, and Portland. All right. And we're trying to figure out, is this relapsing fever? Well, so Schwan, the nosy guy that I am, had to get permission to go up there. And so here I am. My wife took this picture. I'm going into the attic to get nest material to take it back to the lab. And these are Beryllium funnels where you put the organic debris with a hot lamp over it, and if there's live ticks in there, they don't like the heat and the light. They'll move around and they'll fall down the funnel into a jar there. And by doing that, we are able to find the tick. So here's the tick, Ornithodorus hermsii, we got from the cabin of that on the island. And these are spirochetes that we isolated from some of the patients. We got patients' blood sent to us. I put the blood in mice. They grew well. Then we took the infected mouse blood, put it in a particular medium. The spirochetes grew up, and we analyzed them. So we were able to find the spirochetes and the ticks, confirmed that these people were actually infected with this bug, identified it with molecular techniques, and we found the tick vector. So for us, that was pretty interesting. We were able to nail down that Flathead Lake was a newly identified endemic area for tick-borne relapsing fever. So then I went back up with the state epidemiologist and Lake County nurse and talked with some of the lot owners. So here I am over here. This guy started it all because he's the one that got there early and went up to try to clean out the attic, and he's the one that got everybody sick. All right? And I'll tell you why in just a second. But well, I'll tell you now. So what he did was, it turned out, by disturbing the nest material over 2 bedrooms, the ticks fell through the ceiling cracks into the 2 beds in the bedrooms. And the 5 people that slept in those 2 bedrooms got sick. The 16 other people that slept in other places did not get sick. All right? So this guy was doing a good deed, but he got sick, and so did 4 others. All right. Well, there's the gal sitting over here off the screen listening to this. And here she is. Some of you in here might know her. I don't know. It's a small world in western Montana. Well, 2 years, so at that Lot Owners Association, I told the group what had happened and what they need to do if they get sick after being out there. And sure enough, it was another Sunday night late. I got a call from this lady's sister who just flew back to Albuquerque, New Mexico. Interestingly, she's an FBI, the sister's an FBI agent, all right? And she was sick, and she said her niece and nephew were sick, these 2 kids. Do they have relapsing fever? So I always thought, well, I'm not a doctor, but they certainly could. I think, obviously, you need to have them checked out. Well, the mom was already on her way to St. Pat's with the 2 kids. The 2 kids were sick. And she went into the ER and said, my kids have relapsing fever. My kids have relapsing fever. And they said, right. All right? She said, look at the blood. Just what I told that group out on that veranda. Look at the blood. So they disappeared. They came back about 20 minutes later, and the doc said, you're absolutely right. Both your kids have spirochetes in their blood. All right? So in the paper, there was a write-up for another reason here, but you can see it over here. It says, the mother, Linda, not only rushed the children to the hospital, she helped the doctor diagnose the illness. All right? But within 12 hours of onset, these kids were properly diagnosed and treated, right? Which is the goal. That's a kind of a gold standard. If the gold standard is not to get sick at all, of course. All right. The reason this wound up on the paper was that in a previous article in Missoulian, the journalist Stromness suggested that you only get this infection in rat-infested cabins, and this cabin, according to the family, was not rat-infested, so it stimulated a further discussion, in the newspapers. Anyhow, this gal is a senior at Big Sky High School, and she was going to work with me this term to develop a public health brochure for relapsing fever, but she got a better deal to go to South America, so she left me for South America. Yeah. But what was interesting is both those kids got infected, and their aunt got infected. We got the blood, we isolated the spirochetes, we genetically typed them, and one kid, and they all slept in the same bed, all right? The 2 kids and the aunt all slept in the same bed. But one kid got genomic group 1, and the other kid got genomic group 2, and the aunt got genomic group 2. So right in the very same bed, there were spirochetes that were genetically distinct from each other, which to us is interesting. And it generated a whole bunch of work in the lab and in the field. You know, how can that happen? You know, can a single tick have both? As, you can come up with all the combinations. So I finally got a good student and worked with Carrie Forsman in wildlife. And from 2008 to 10, Tammy Johnson did her PhD project up at Flathead. And we sampled wild horse, Melita, Polson Big Arm, and then over at the Yellow Bay Research Station in Upper Yellow Bay. We tried to do Cedar Island, but that was a waste of time. Anyhow, so we set up a 3-year field study to see what animals might be maintaining the spirochetes and ticks at Flathead. And here's Bob Fisher on the right and Tammy Johnson on the left. And they're fully geared up with personal protective equipment. Anybody know why they might have to wear this?
So they don't get bit.
Huh?
So they don't get bit.
Well, there's another reason.
You get shorts on.
Huh? Yeah, mice. Hantavirus in deer mice. All right? So we caught a lot of deer mice, and they're the reservoir for hantavirus. So the protocols to handling deer mice require these full body suits, shields, face respirators, etc. When you're handling chipmunks and squirrels, you do not have to dress like this. But when you're processing deer mice, you do. And so that's Tammy, the PhD student. She's now an ASM fellow at CDC in Fort Collins. And Bob Fisher's now been, since I retired in January, Bob's been transferred to the Laboratory of Virology. Here's Tammy again. We had a field trailer set up. This is Lisa Wolf. She was a veterinary student. Now she's doing a PhD back east. And Ben Kreischt's, he's now doing the PhD down at Colorado State. All right. So here's an island, wild horse, and then Melita. Basically, these islands are loaded with animals that are supporting ticks and spirochetes. All right. So Melita is owned by the Boy Scouts of America, and here's the results of our lab work. We capture, you don't have to worry about remembering all this, but basically we caught 11 species at all total of the sites, and a quarter of them had antibodies showing that they had been infected previously. And just under 3% of them had an active infection at the time we caught them, which meant we caught them, we collected blood, made a smear, spirochetes in the smear. Okay. And this is my harangue about serology over direct detection we can forget. All right. So in this Montana study, really we found that the pine squirrels were the most important vertebrate host, probably for supporting the ticks and the spirochete. So you can see that almost, 80% of the pine squirrels we looked at had been previously infected. That's a whoppingly high seroprevalence, whoppingly high. The other thing is we found infected squirrels on wild horse, Melita, and Yellow Bay. And we found infected ticks as well. That's the next slide, I guess, yeah. So we also tried to collect ticks. These ticks are not easy to find. They're really hard to find. And you can collect them with dry ice traps, and I'll show you that in just a minute. But we didn't, for 3 years, we only got 31 ticks, which sounds pitifully low. But at least in the lab, 13 of these 31 transmitted to mice, and we were able to isolate the spirochete and genetically type them. And basically nailed down that Flathead Lake area is an endemic area for tick-borne relapsing fever. All right, I don't know if I can do this with you guys, but I'm going to try to play a game. Now we're here July 22nd, 2013. There's some folks in here that had to play this game with me before, so it won't last long. Okay. 55-year-old male, resident, east of Corvallis, Montana, acutely ill, sent to the ER in Marcus Daly, and then transferred to St. Pat's, and the attending physician is Joshua Christensen, who's we've been working with. Okay, so that happened in July of last year, and then I got a call from Dr. Christensen saying he had a case of relapsing fever, and could we identify the spirochete? I said, sure. I mean, that's what we do. We can do that. And not to brag, we can do it better than anybody else. That's just what we do. All right, so with the patient's permission, he sent a blood sample, and here's where I made a thin smear, and then fixed it with acetone, and then stained it, or actually in this case methanol, and then stained it with an antibody that is specific for these types of bacteria. And then when you look at them, and they're bound to a fluorophore, and then when you look at it with a UV light fluorescent microscope, the spirochetes just light up with fluorescence. And so here they are. You can see 3 or 4 spirochetes here that were in the patient's blood. All right, well, that's cool. This guy had Borrelia hermsii in his blood. And he was really sick. He was hospitalized for 5 days. The guy was sick. All right, so this is what I would want to know, right? Where did the patient get infected? He lives in Corvallis. So what would you want to know?
Has he taken any trips recently?
Travel history. There we go. Bingo. But you didn't take my class. I'm sorry, never mind. All right, so travel history. So we went over that. This guy works in Antarctica. He had been into Spain and Turkey and all over the place. He'd just done a motorcycle ride through Glacier, but never got off the pavement, supposedly. He was all over, but for the weeks prior to his onset of illness, he did not leave the Bitterroot Valley. So this is then when I got excited, right? Yeah. The patient moved part of a wood pile in his backyard on July 11th. And I talked to him. We got permission to talk to the patient, which sometimes is tricky. This guy was very cooperative. He's a scientist, NASA scientist, so it's really fun working with him. There are chipmunks on his property. Used to be pine squirrels on his property. All right, but 5 days before he came ill, he messed with a wood pile right behind his back door. So we asked if we could do an on-site investigation. And he said, you bet, no problem. So, and he has some big rock outcrops on his property, and it's a pure stand of Ponderosa pine, second growth. It was all cleared in the 30s, and now his property is rocks and pure stands of secondary growth pine. And chipmunks, we didn't actually catch any squirrels, but you can see there's 2 types of traps here, a small Sherman trap and a larger tomahawk trap. And there were brush squirrel, rabbits running around. But here's Bob and Brandy setting up our little field lab out on the fellow's property and getting ready to sample the rodents that were captured. All right, so here's a chipmunk in a bag. It came out of its trap, and it's getting anesthetized. All right, so we've got an anesthetizing cone here. Here's the head. Here's the blood. Here's an empty microscope slide waiting to get a little blood to make that spear so we can look for spirochetes. And then, well, let me just go back and say, that chipmunk was infected. All right? That chipmunk was infected with spirochetes. That made my month. And I put it into the mice. We isolated it, and it was genetically, base for base, identical to what we got out of the human patient. Identical. All right? Then, of course, he said that he probably was bitten when he was messing around with his woodpile. So here's the woodpile, and we put a hardware cloth down, and then there's a big block of dry ice underneath this styrofoam cooler. Now, when dry ice sublimes, it releases carbon dioxide, and that's a bait for many blood-feeding arthropods. So in this and so then we kind of got it also protected. So there's dry ice under here, and there's carbon dioxide gas coming out like this. And the next morning, voila, we got the beast. All right, we actually found the tick vector right in the woodpile. And we've got 10 ticks out of that woodpile, and 4 of them transmitted when they fed on mice in the lab. All right? And somewhere here, well, I'll get to that. I can skip one slide. So this is pretty cool. We got the bug from the patient. We got the bug from a chipmunk. We got the right tick vector in his backyard. We got the bug from the ticks. Yeah? That's what you want to do if you want to nail down where someone got infected, all right? So we're absolutely convinced that he was infected on his own property. Not when he was sleeping, and what he has is a very nice house, but by messing around in the woodpile during the day. And there were cut cones in there, and we actually found ticks in the woodpile. Well, he didn't want any ticks in his woodpile, all right? So we went down and bought over-the-counter acaricides, and he bought the tarpaulins, and we totally covered that woodpile and then set off these experimental bombs that would kill the ticks. And then he got rid of the woodpile. We also found some serological evidence from some of the other animals there that are positive. So a blood sample can be looked at in an immunoblot. But here it is right here. Here's a negative animal serum, and here's a positive. That's all I'll say, but there was evidence of other animals having been infected on his property. All right. So this is kind of cool for me. Here's Hamilton. Here's the lab. And this is the area where the ponderosa pine are, about 7.5 miles. All right? So we've got an endemic focus of this disease 7.5 miles from the lab. And people have been working on ticks there for 100 years. And we've nailed it. And what's kind of cool to me, it's only 3.5 miles from that stock farm, kind of a fancy place. All right? So this is some of the ponderosa pine and the golf course here in the Ritzy stock farm. So I bet they've got the potential for relapsing fever at that stock farm. All right. So just to summarize this, I think I've already said it. And then, yeah, I've said all this. We got it from the patient. We got it from the chipmunk. We got it from the ticks. So the Bitterroot Valley is also an endemic area for tick-borne relapsing fever. Now, I know we're getting close, but I think I can end this in 10 minutes because we also are very interested in this disease in Africa. All right? And actually, the first observations of tick-borne relapsing fever were made in Africa in 1903-4-5 in East Africa. So our institute has a center of excellence for research in Mali, West Africa. So here's the African continent. Mali is landlocked here in West Africa. And we've sampled 20 villages over about 7 years. I've been over there 8 times where we travel across and sort of do what we do in Montana, but do it in Mali to see if there's a risk of relapsing fever there. And the first diagram of showing a human infection from Africa is shown here. I'll go over it really fast, but it's pretty cool. Here's a chart showing calendar days, a line for normal body temperature, and then here's the temperature curve for this patient over a few months. And at that time, they took blood samples and they found spirochetes in the blood then, but not then. Then, but not then. Then, but not then. So again, getting back to what I mentioned about the changing antigenicity of these bugs over time so the bugs can repopulate and cause an illness in people. Now, what I haven't said yet is these bugs didn't evolve to make people sick longer. All right? They evolved to be able to continually repopulate the blood so that they're available for the tick vector to get them when they feed in 15 minutes, all right? It's a beautiful adaptation to prolong the availability of these bacteria to be acquired by their tick vector. They didn't involve this mechanism to make people sick repeatedly, all right? But when people get infected, they get sick repeatedly, all right? Anyhow, this was 1905, and one of the guys doing the study got sick, and this is his fever curve right here, all right? So our interest in West Africa is relapsing fever being confused with malaria? And it probably is. And this is just a quick list of some of the signs and symptoms. But when people have an acute recurring illness in Africa, the assumption is it's malaria. All right? Relapsing fever has not been on the radar screen, and that's what we've been trying to do. Get it on the radar screen so people get treated appropriately. So just a little bit about Mali. 90% Muslim. This is the largest sand, mud mosque in the world. In Jenny, it's a World Heritage Site, UNESCO World Heritage Site. It's an amazing structure. The 2 major rivers that flow through and give the life to Mali, because it's landlocked, and the Sahara Desert is the 2/3 to the north. These 2 rivers really provide what people need to live, the fish, the irrigation, the vegetables, etc. Most of our work began to focus in on Dogon country in the Bandiagara area, and these are Dogon storage huts. I just love the culture over there. It's just fascinating. And when we go into Malian village, the first thing we have to do is meet the chief, meet the elders, and then of course I don't speak French, I don't speak Bambara, I don't speak Dogon. So our Malian colleagues set the stage, explain why we're there, what we want to do. We want to trap animals, we want to look for ticks, and they never say no. All right? They're always gracious and want to help us do our job. All right. And here I am just explaining to some of the folks how these live traps work, where they've got a false door and then the animal walks in and they push the floor down and the door shuts on them. All right. And this is not a very impressive setting, but we did a trapping in an NGO a Canadian-supported garden, community garden, out in the middle of nowhere. and where they grew onions. And the rodents were coming through these fences and just eating their onions down. I mean, just grubbing down their crops. And we caught so many rats there. If we ever went back again to that place, I'd like to have a project to take them 1,000 rat traps or something. But anyhow, we caught a lot of animals right there. Yeah, here's Nafo, one of our primary collaborators. This is not the way you open a trap if you think you have an animal in it. All right, it's just not the way you do it, but you know, some things you just can't get across. All right, so part of the problem with the work is knowing what you're catching. If you want to find out what animal is the primary vertebrate host for the spirochete, you need to know what you're catching. And in Mali, that wasn't particularly easy. All right, so and it's easy if you catch the world's smallest mouse. Adams knows this, so you know he's so. All right, and it's not too tough if you catch the world's largest rat, right? So this is Chrysotomes. You've just seen the world's smallest rat and the world's largest rat. But what happens if this is your morning catch? All right, then you got your hands full. All right, and this isn't catch and release. All right, sorry if there's any out there don't like this. Nobody over there that wants us to trap wants us to let those rats back in their kitchen or their bedroom or their grain storage bin, whatever. You know, we have to euthanize them, and we work them up. We take measurements. This is one species, the mastomys. So every animal that we captured over the years got photographed with a ruler, body weight, tail length, etc. Skin sample for DNA. This is a shrew. The shrews over there are huge, and they stink. All right, and these things are just running around the bed. You know, the beds on the ground. I mean, these people live with a lot of vermin. All right, so we set up little roaming labs, just a plastic bench, and we're anesthetizing, collecting samples, euthanizing, and we always have young folks interested in what we're doing. This picture I like because it's kind of dark, but I didn't know this young man was watching me, but I'm preparing heads for voucher specimens. And then when I got them home here, I was able to get them clean in my garage. I have beetle colonies and I clean them. And now these are all in the Smithsonian Institute as voucher specimens. We actually have specimens in the Smithsonian that are there to base our identifications on. So if we say the Relapsing Fever spirochetes in Mali are being maintained by this particular species of rat, we can say, if you don't believe us, here's the DNA data, and you go to the Smithsonian and say, we want to look at Schwan's number, RML 50 through, you know, whatever. And so, and believe it or not, these were the first mammal specimens from Mali that the Smithsonian ever received. Anyhow. All right, so I'm going to wrap this up real fast here, but that's what we were looking for in the blood smears. These are spirochetes. This is, for us, the holy grail. We like to see the organism in an infected animal. So if you can, hopefully you can see them. Here's a spirochete, here's a spirochete, here's a spirochete. This is the cause of relapsing fever in Mali. And so we found, five villages where the animals were actively infected the time we sampled. We also did serology where we take blood samples and look for antibody. That's all I'll say here. But you can see some positive animals here, negatives here, because the animals have antibody that bind to all sorts of the proteins that comprise those spirochetes. And then we found animals with antibody in 14 of the 20 sites. So we think that these spirochetes are circulating widely across southern Africa. Now, the famed city of Timbuktu is about right here, all right? And this is all Sahara Desert up to here. Yeah. And then, of course, there's a third component. There's rats, there's spirochetes, and then there's, huh? Ticks. Ticks. We want the ticks, all right? So we have to go looking for ticks. And so what we used was a Sears Roebuck leaf blower, all right? And we reversed the hoses on it, so rather than blow, we suck. So here's the nice Sears Roebuck leaf blower with the attachments that are going to now suck out of the burrows in the people's houses. And here we go. This is a gas-powered machine. The hoses are going in, down the burrow. These animals are living right in the people's houses. and then we suck out the dirt, hopefully the ticks. We put them in screens. This is Usman Maega, another important collaborator. And voila, that's the tick we've been looking for. All right. So we were able to find about 750 ticks. It took, I don't know, umpteen permits to be able to bring them back to Montana and feed them on mice, and they transmitted, and we isolated spirochetes. So this is about the end here. I've had 8 trips to Mali, sampled 20 villages, a lot of animals, seropositives, actively infected, ticks infected, and identified the spirochete as Borrelia crossideri. And if the politics calm down and we're able, we want to go back and set up a clinic where we can actually then start monitoring human infections to determine what is the incidence of this infection in humans in Mali. So, I'm done. On time, right?
Good.
All right.
How did you come across the diagnosis on a dog? Because it doesn't seem like the vet would naturally check for this, so it seems very interesting that you have to do it with dog diagnosis.
Yeah. So, for a lot of these things, we work with other people. For the dog, it was sort of a circuitous route. We had been working on infected dogs in Texas with a different species of spirochete. We've done that over quite a few years, where another species of Borrelia can cause illness in dogs in Texas. All right, and those papers were published. What happened was someone in Seattle at a veterinary clinic had a dog brought in, and there was a identification of spirochetes in the blood. She thought it was leptospirosis initially, so she sent the sample down to a diagnostic lab in Texas, and they tested it for leptospirosis that was negative. They did a molecular test to try to identify the spirochete as lepto. It didn't work. And somebody else said that she ought to contact me. So she contacted me, and I said, do you have any blood left? Well, it's down in the diagnostic lab in Texas. So I asked them to send me the sample. It was just a little bit of serum left. But when I got it, under the microscope, there were live spirochetes. So that makes me happy. All right. So I put that into a mouse. The mouse became infected. The spirochetes grew to a high level. I was able to isolate it. And we identified it as the right species, what genomic group, and then we worked with the vets to write it up. This dog got infected over near Lake Schwan, is it? Schwan? In Washington? Yeah. Yeah, the dog had stayed in a cabin, just like people do. This dog had slept in a cabin over in that lake area, high elevation, and got sick, was taken to a regional vet, and then that vet sent the sample and write-up to this diagnostic lab over in the Seattle area. Yeah. Right. Mm-hmm. Yeah. So we got in on it sort of late in the game, but they hadn't worked it up or identified it yet. The dog was treated and is okay. Yeah. But the dog was sick, and I think on wild horse, I bet there are dogs getting infected with Borrelia hermsii on wild horse. And we had hoped to actually do a survey on dogs for all the dogs that go there, but there's just too many things to do, and it didn't happen. Yeah, maybe it can still happen, but I probably won't do it. Yeah.
That retired thing.
Yeah.
Like you're moving away from it.
Yeah, it hasn't worked yet. Yeah. Yo.
So could you talk a little bit more about the disease? You said that if you give antibodies, it'll come back again and again. Do you just administer antibodies and then they're done? They don't have the disease anymore? Or is it going to be a thing that's chronic throughout the rest of their lives?
Yeah, I didn't explain that very well. First, this is not treated with antibody from another person who's been infected and has a serum sample. Some days in the old days, people would take convalescent samples from people who had antibody to something and administer it to help protect or reduce help the outcome of an infection. Is that what you're asking?
No, just antibiotics. Are they?
Oh, antibiotics.
Yeah.
Oh, I'm sorry. Sorry. Okay, so these spirochetes these infections are easily treated with antibiotics. Tetracycline, penicillin, amoxicillin. I had a call from a woman 3 nights ago whose daughter got sick in Colorado. Her daughter got ceftriaxone IV. That's a rather rigorous treatment for a kid, but this girl was really sick, and and in Africa sometimes they treat it with only one pill. In North America it's typically 10 to 14 days oral antibiotics, but I'll tell you that it only takes 1 or 2 doses to pretty much clear the infection.
Okay,
No.
so it doesn't come back after it's treated with antibiotics?
No, no, it does not. The main thing that can happen with the onset of treatment is a reaction called the Yarish-Herxheimer reaction. And the antibiotics will kill the bacteria, lyse them, and then all of these components can be toxic. When you have 10 million spirochetes per milliliter of blood in a large body, and then treat that with an antibiotic that will kill all of those bacteria, millions and millions of bacteria are killed and lysed, a person will go into a toxic shock. And that can kill you too. People have died from the treatment. Not so much in North America, but in East Africa that happens unfortunately more than it should. So people when treated should be treated under observation, kept in the hospital room, and if they have that reaction, you want to get them through it and recover from it before they're released. I mean, I've talked with, when I worked with folks in Deaconess in Spokane, I mean, they would treat these people and put them in their car, and these guys were going home, they drive off the road. They get this reaction, and off the road they went. They were lucky they didn't kill themselves in an automobile accident. So, but antibiotics work. Yeah.
How often do you get toxic shock from that?
It's, I think it's more the norm than not. I think most people, so it depends on how dense the spirochetes are at the time of treatment. That's why I personally think that rather than treat someone when the spirochetes are at a peak, the body's going to crash them down to an almost undetectable level. And I think that's when they should be treated. I'm not a doctor, but I think that's when they should be treated because it would greatly reduce the chance of a severe shock reaction. But people don't do that. If someone is diagnosed and they have, like I said, 10 to the 8th spirochetes per mil of blood, they'll be treated then. And then, the stimulus for that shock is just compounded by log number increases the number of bugs. So I can't tell you. I think it's going to vary. I mean, it'd have to be a controlled experiment where you treat people with the same densities of bugs. Yeah.
Are there any studies that would show the incidence of Lyme with recurrent fever at the same time of infection?
Could you find dual infections?
Yeah, comorbidity.
Well, there's plenty of dual and triple infections reported in the East because a single tick that's a vector of Lyme disease is also the vector for the anaplasmosis bacterium and the babesiosis protozoan. And so there have been records of where a single Ixodes tick has transmitted to one patient the Lyme disease spirochete, the anaplasma bacterium, and the Babesia parasite. That can happen. But the relapsing fever and Lyme disease areas really ecologically don't overlap. So there's a separation ecologically and geographically, all right? There's no true relapsing fever east of the U.S. throughout the endemic Lyme disease region of the East. And where Lyme disease occurs primarily on the western coast, it's lower elevation coastal areas where relapsing fever doesn't occur. So, that's about all I can say. It could happen, but you'd have to have, it can't happen by the same tick, all right, because you've got 2 different types of ticks that are vectors for these. So you can't be duly infected by one tick bite and get relapsing fever and Lyme because it's totally different ticks that are transmitting those bacteria.
Well, I want you to join me in 2 things. First, to thank our speaker for tonight, and secondly, to hang in there while I tell you about next week. So first, thanks very much, Tom, for really